and I want to introduce Jason Sheltzer. Jason is an assistant professor of surgery and received his PhD from MIT, where he worked in the laboratory of Dr. Angelica Amon in the Koch Institute for Cancer Research. After completing his PhD, he established his own research group as an independent fellow at the Cold Spring Harbor Laboratory. The Sheltzer Lab is broadly interested in understanding the genomic changes that drive cancer progression, particularly aneuploidy, which is found in more than 90% of human tumors. Additionally, they're working to identify genomic alterations that create druggable therapeutic vulnerabilities in cancer. They have recently discovered the first ever selective inhibitor of the kinase CDK11 and are developing CDK11 inhibition as a new strategy to treat malignancies. Without further delay, Jason, all yours.
Right. Thanks so much for the kind introduction. So I'm very excited to be able to share with you today some research my lab has done about off-target activity of cancer drugs undergoing clinical trials. These are my disclosures. And this project really comes from a journal article that I read a few years ago that had a statistic in it that I found to be just absolutely shocking. If you look at all drugs that enter clinical testing in oncology, 97% of drug indication pairs that enter clinical trials fail during that testing and don't end up receiving FDA approval. And this 97% failure rate for oncology drugs is the highest of any field of medicine. So more cancer drugs fail than psychiatric drugs or endocrinology drugs or infectious disease drugs or anything else. And if you look at the proximate causes for trial failure, the most common immediate causes that drugs run into are toxicity and limited efficacy. That is, the drugs have too many side effects for patients to safely take, or maybe the patients can safely take them, but they have limited efficacy and they don't actually shrink a patient's tumor. And while these are kind of the proximate causes for oncology drug trial failure, I think the underlying reasons why so many drugs run into these problems isn't very well understood. And today, I'm going to share some evidence from my lab towards one potential explanation for this high failure rate. And the hypothesis that I'm going to argue for is that many drugs are entering clinical testing in oncology with an incorrect understanding of their mechanism of action. And I think this mischaracterization of cancer drugs may be one factor, by no means the only factor, but one factor that contributes to this extremely high failure rate for oncology therapies. So in my lab, we've been interested in using genetic approaches to investigate the mechanisms of action of different experimental cancer drugs. And by searching through the current literature and looking on clinicaltrials.gov, we put together a list of 12 different drugs targeting 7 different cancer-related proteins that we were interested in studying. These drugs have been used in about 30 different clinical trials targeting several hundred cancer patients. So 6 of these proteins are reported to be cancer genetic dependencies. That is, the function of these proteins is reported to be essential for the growth and proliferation of cancer cells. For instance, PAC4 is a kinase. It's been reported that PAC4's kinase activity is essential for the growth of colon cancer, lung cancer, breast cancer, and a few other cancer types. And because of that genetic data concerning PAC4, that motivated Pfizer to develop a small molecule PAC4 inhibitor, PF-3758309, which they then entered into clinical testing. Caspase-3 is a little bit different. I'm going to talk about caspase-3 separately. So we were interested in testing the mechanism of action of these drugs and seeing whether they killed cancer cells through the inhibition of these proteins. And as a first step towards this process, we wanted to confirm that the proteins these drugs were targeting were truly cancer genetic dependencies. That is, they were essential for cancer growth. And to investigate this, we set up a Crispr competition assay to see what happened when we knocked these genes out. To do this CRISPR assay, we transduced cancer cell lines with Cas9, and then we transduced them a second time with a guide RNA co-expressed along with GFP. This would then create a mixed population of GFP-positive cells that had the guide RNA and caused mutations in the target gene, and then untransduced non-fluorescent cells. We then measured the percentage of GFP cells over time. If the percent of GFP positive cells decreases over time, that tells us that whatever gene the guide RNA is knocking out, it must be required for cancer growth because the GFP positive cells are dying. In contrast, if the percent of GFP positive cells stays about the same, then that's evidence that whatever this guide RNA is targeting, it isn't important for cancer growth because these GFP positive cells can grow just fine. So that's what the assay looked like. We designed and cloned multiple guide RNAs targeting each of the putative cancer genetic dependencies we were interested in studying. And then we did a bunch of competition assays. And this is what one of these competition assays looks like. So here we're in MDA-MB231 cells, a triple negative breast cancer cell line. As negative controls, we have guide RNAs targeting non-essential loci, ROSA26 and AAVS1. Guide RNAs targeting these genes exhibit no dropout. As positive controls, we have guide RNAs targeting the essential replication genes RPA3 and PCNA. Guide RNAs targeting these genes, which are required for DNA replication, drop out between 50-fold and 200-fold over 5 passages in culture. We then looked at the effects of guide RNAs targeting each of the putative cancer genetic dependencies that we were interested in studying. And we were really astounded when the guide RNAs targeting these cancer drug targets exhibited no dropout whatsoever. These guide RNAs behaved exactly the same as guide RNAs targeting known non-essential genes like ROSA26 and AAVS1. And this was incredibly surprising to us because right now there are patients who are receiving anti-HDAC6 therapy and anti-MELK therapy and anti-PIM1 therapy based on the belief that these proteins are required for cancer growth. But this experiment suggests that in these experimental conditions, in this cell line, we can eliminate these genes without any effect on cancer whatsoever. So this is what it looked like in one cell line. We ended up repeating this assay in 32 different cancer cell lines for more than a dozen different cancer types. And in each of these experiments, we got the same result. There was no dropout of the guide RNAs targeting these drug targets, and there was no evidence that any of these genes were actually a dependency in any cancer type. So this made us take a step back and think, well, is there something that could be going wrong in this assay? Could we be, doing something incorrect here? And so we thought, well, maybe with CRISPR, we're generating heterozygous mutations, but not homozygous mutations. Maybe we're introducing mutations into these genes, but we're not really knocking out the total protein. So we thought, okay, instead of doing this population-based approach, let's make single-cell derived knockout clones and be as sure as humanly possible that we were really eliminating 100% of the target protein. So we did that. We used a 2 CRISPR guide RNA strategy where we designed a guide RNA targeting an upstream exon and a downstream exon so that we could physically cut the gene out of the genome and there would be no protein left. So we sorted single cells that were double positive that picked up both guide RNAs that we transduced in. And then we verified target knockout using 2 independent antibodies. So for instance, one gene we were interested in studying is MAPK14. This is the gene that encodes the kinase P38 alpha. We generated knockout clones and we verified complete target knockout using one antibody and then verified it again using a second antibody so that we could be, as sure as physically possible that we had truly eliminated all trace of these putative cancer drivers from the cell. However, when we tested the fitness effects of these knockout clones, we got exactly the same result that we got from the competition assays. Knocking out these putative cancer genetic dependencies had no effect on cancer growth. So here, for instance, is a proliferation assay in a melanoma cell line. We have 3 MAPK14 knockout clones and then 2 control ROSA26 clones. And these MAPK14 knockout clones grow exactly as well as the ROSA26 control clones. We could also put these cells in soft agar, challenge their clonogenic ability. We saw no difference in clonogenic ability either. These knockout cells grew just fine. So to sum up a whole bunch of data that I don't have time to show you, we ended up eliminating all 6 different cancer driver genes that we were studying in at least 3 different cancer types each, and there was no fitness effect whatsoever that we could discuss. So this was a really strange finding to us, and it made us try to figure out what was going on. So we were looking at the targets of 12 different anti-cancer drugs in various stages of clinical development. And we looked at these drug targets with multiple different CRISPR techniques. We did CRISPR competition assays, we made CRISPR knockouts, but concordantly, they both showed that we could eliminate these genes without a detrimental effect on cancer proliferation. This then raised the question, well, why were these genes believed to be cancer essential in the first place? And when we looked into the literature on these genes, we found that two main lines of evidence had identified these genes as cancer essential initially. The first line of evidence identifying these genes as cancer essential were experiments done using RNA interference. The second line of evidence were experiments done using small molecule drugs, many of which had then gone on to enter clinical testing. So we wanted to see if we could backtrack a little and understand why we had come to such a different result than these previous experiments done using RNAi and small molecule drugs. So I'll first show you what we learned when we looked at some of the prior RNAi experiments. So this is an RNAi experiment published in the literature a few years ago that had identified the kinase PAC4 as essential for the growth of colon cancer cells. In this experiment, the investigators took siRNAs targeting PAC4, they introduced them into HCT116 colon cancer cells, and they found that the siRNAs decreased colon cancer cell survival. And data like this motivated Pfizer to enter a PAC4 inhibitor into clinical trials. We had found no fitness effect when we had knocked out PAC4 using CRISPR, so we wanted to see if we could recapitulate this result that had been published using RNAi. Two of these siRNA constructs were commercially available, and we had HCT116 cells growing in my lab. So we purchased these siRNAs from this prior publication and then tested them in our cells. We transfected these siRNAs, the same from the prior publication, into HCT116 cells, and we could confirm by Western blot these siRNAs decreased protein expression as expected. And we did a cell survival assay, and we could confirm that they killed HCT116 colon cancer cells exactly as had been reported. However, using CRISPR, we were also able to generate a PAC4 knockout clone in this exact same cancer cell line. So here we had a PAC4 knockout clone. You can see there's no PAC4 expression in either the control or the knockdown condition. And then when we did a cell survival assay on these cells, we found that transfecting the PAC4 knockout cells with PAC4 targeting siRNA had exactly the same detrimental impact on colon cancer survival as it did in the PAC4 expressing ROSA26 control cells. So these PAC4 targeting siRNAs are killing colon cancer cells, but their ability to kill colon cancer cells is entirely independent of the expression of PAC4 because they're exactly as lethal in the control cells expressing PAC4 as they are in the PAC4 knockout clones that we generated using CRISPR. So this prior experiment was totally reproducible. These siRNAs killed colon cancer cells, but just the interpretation was wrong because the toxicity of these siRNAs is just entirely independent of PAC4 expression. And this seems to be commonly the case where we test siRNAs and shRNAs in the literature over CRISPR-derived knockout clones. The si and shRNAs may kill cancer cells, but it's just independent of the expression of the gene that they were designed against. The next thing that we wanted to figure out was what was going on with the small molecule drugs, many of which had then gone on to enter clinical testing. And I'll show you what happened with one of those drugs. So PAC1 is a drug that was described a few years ago in a paper in Nature Chemical Biology. And it was developed as a caspase-3 activator compound. So the apoptosis enzyme caspase-3 is normally present in an inactive procaspase state in the cell. And PAC1 was developed to catalyze the conversion of caspase-3 from its inactive procaspase state to its active caspase-3 state, at which point it would then kill cancer cells. And this drug has been entered into 3 different clinical trials. However, this mechanism of action was worked out based on in vitro biochemistry, and no one had described a mutation in caspase-3 that conferred resistance to it, or had assessed the effects of this drug in a caspase-3 knockout cell. So using CRISPR, we generated multiple caspase-3 knockout clones, And then we did a dose response curve examining the viability of wild type and caspase-3 knockout clones in different concentrations of PAC1. So this is what it looked like for two control clones, two clones expressing a ROSA26 guide RNA. PAC1 is a potent anti-cancer agent. You can see it has an IC50 value of around 1 or 2 micromolar. However, when we did the same assay in the caspase-3 knockout clones that we generated, we ended up getting exactly the same drug curve. This drug is exactly as potent in caspase-3 knockout clones as it is in caspase-3 expressing ROSA26 control clones. It has an IC50 value of 1 to 2 micromolar, regardless of whether these cells express caspase-3. So this drug, which entered clinical trials as a caspase-3 activating compound, its anti-cancer activity actually comes from something entirely independent of caspase-3. And this is actually the case for many of the drugs that we studied. So to show you a few more examples, HDAC6 is a histone deacetylase. Celgene has developed HDAC6 inhibitors, siderinostat and ricolinostat. We knocked out HDAC6, but we saw no change in sensitivity to these putative HDAC6 inhibitors. MELK is a cancer-related kinase. Oncotherapy Sciences developed this drug. Novartis developed this drug. We used CRISPR to knock out MELK, and we saw no change in sensitivity to these MELK inhibitory compounds. So to sum up a whole bunch of data that I don't have time to show you, we found that target knockouts conferred no resistance for 12 different cancer drugs that we were studying. We made these knockouts and did these tests in at least 3 different cancer types each. So this kind of leaves us in an odd position. We were studying 12 different preclinical or clinical anti-cancer drugs, and in each of these cases, we found that the reported mechanism of action was actually incorrect. This then raised the question, well, if these drugs are killing cancer cells at nanomolar or low micromolar potency, how is it they actually work? What is it they're actually targeting? We wanted to see if we could figure out how they were actually functioning. And we've had the best success so far with one drug called OTS-964. This is what the drug looks like. It was described in a paper in Science Translational Medicine a few years ago as an inhibitor of a kinase called PBK, which is also called TOPK in the literature. But using CRISPR, we knocked out PBK and we saw no effect whatsoever on sensitivity to this compound, telling us that this drug, OTS-964, must have some other cellular target. To see if we could figure out what this drug was actually doing, we used a genetic-based approach. For this approach, we took highly mutagenized colon cancer cells, HCT116. They have a very high mutation rate because they're microsatellite unstable. And then we exposed these drugs to a nearly lethal concentration of OTS-964, such that about 99.9% of cells on the plate were killed. However, there were a few stragglers that remained, and we kept these cells in the drug for a period of weeks until these cells were able to grow and form little microcolonies. We then subjected these cells to whole exome sequencing, and when we did sequencing on the resistant clones, what we were hoping to see was a mutation that blocked whatever it was this drug was actually targeting. Maybe these cells could survive a lethal treatment because they had some mutation preventing drug binding to whatever OTS-964 was actually doing. So when we did whole exome sequencing on these clones, we were really excited to see that every clone that we looked at had the same mutation in it. Every drug-resistant clone had a mutation in the cyclin-dependent kinase CDK11. They had a glycine to serine substitution right smack dab in the middle of the CDK11 kinase domain. So this immediately suggested to us that maybe this drug, which had been developed as a PBK inhibitor, was actually functioning through inhibition of CDK11 instead. One potential limitation to this is that, well, there actually isn't a precedent for this. CDK11 hasn't been previously drugged. So we wanted to see if this mutation actually had anything to do with sensitivity to OTS-964. In order to do that, we wanted to see whether this mutation that we discovered in the resistant clones was actually sufficient to confer resistance to OTS-964. To test this, we used a CRISPR knock-in strategy where we introduced this glycine to serine substitution that we recovered in drug-resistant cells. We knocked it into drug-naive cancer cells and then tested its effects on OTS-964 sensitivity. This is what it looked like. Here we have 4 different cancer cell lines treated with a lethal concentration of OTS-964. With a negative control guide RNA, or if we just cut in the CDK11 gene, we have no cancer cell viability. But if we introduce a repair template that includes the glycine to serine substitution, then we can restore viability in the presence of an otherwise lethal concentration of OTS-964. So this tells us that this mutation is in fact both necessary and sufficient for resistance to this compound. We then followed this up with some biochemical assays. We confirmed that OTS-964 inhibits CDK11 with an IC50 value of around 40 to 50 nanomolar in vitro. And we did a cell-based target engagement assay using mass spectrometry. We found that 100 nanomolar treatment with OTS-964, it didn't bind to hundreds of other cellular kinases, but it caused about 70% binding site occlusion for CDK11 and only CDK11. So from this work, we concluded that by profiling a mischaracterized anti-cancer agent, we were actually able to serendipitously discover the first selective inhibitor of CDK11. So to sum up what I've told you so far, we're kind of operating in a space in which the vast majority of new therapies that get tested in human patients in oncology don't end up working. And we put together a collection of these drugs to study, and one thing that we found while studying them is that many of these drugs have actually been designed to target proteins that have no detectable role in cancer growth. Furthermore, while these drugs do kill cancer cells, they largely kill cancer cells through off-target effects rather than through the target that they were initially designed against. And I think that this can increase the burden of side effects and decrease the efficacy when some of these drugs are actually used. We don't truly understand how they're working or where their anti-cancer activity comes from. I think this conclusion has a number of important considerations and caveats, though. For instance, there could be unrecognized cell type specificity. We did these competitions in 32 cancer cell lines. We generated knockout clones in 3 cancer types each, but it was, physically impossible for us to test every subtype of leukemia or every subtype of kidney cancer in existence. And so we can't fully rule out some unrecognized cell type specificity that hasn't been reported in the literature on these targets. Secondly, we specifically tested the hypothesis that these proteins are required for cell autonomous cancer growth. That is, cells going from 1 cancer cell to 2 to 4 to 8 and so on. And this had been reported for each of the drugs that we had studied. However, if it turned out that, say, PAC4 had some role in angiogenesis or in immune evasion or some other non-cell autonomous process, that wouldn't be ruled out for the cell autonomous proliferation focused assays that we've done. I think a third important consideration is, while our data suggests that these drugs are promiscuous and may have multiple targets in the cell, just because a cancer drug is promiscuous doesn't necessarily mean that it will fail in the clinic. There are a number of drugs like sunitinib and sorafenib, which do have multiple targets in the cell. And so just because something is promiscuous doesn't necessarily mean that it will fail. However, I think that if our goal in cancer biology is to kind of reach a plateau of targeted precision medicine, where you sequence a patient's tumor, you identify the mutations and amplifications and alterations, and then design a drug cocktail based on that particular genetic profile, in order to get to that level, I think we need to have a really good understanding of what drugs do and how their anti-cancer activity actually arises. And what we'd suggest is that preclinical genetic validation, particularly using Crispr instead of Rnai, may help us get genetic insight into how anti-cancer drugs work and may decrease the number of investigational drugs that enter clinical trials but end up failing during clinical testing. So this is work that was done by my group, in particular 2 really talented students, Ann Lin and Chris Giuliano. I'd like to acknowledge the funding, and thank you so much. I'd be happy to answer any questions that you have.
Thanks very much. I thought that was really great. I think one of the things we're all aware of is that when we combine drugs, that the toxicity goes way up. And of course, much of the reason for that is that many of these drugs are promiscuous and are doing much more than what we need them to do. There was a question a minute ago. From Jeffrey Townsend, how were the original 12 drugs selected and assembled for investigation?
Yep. So I didn't have time to discuss that extensively in this talk, but what we were interested in, our underlying hypothesis is that the gold standard for knowing a cancer drug's mechanism of action is the identification of a mutation that confers resistance to it. The classic example here is Gleevec and the mutations in BCR-ABL that block Gleevec activity. And our thinking was that drugs that lacked that level of genetic validation were less likely to be acting through an on-target mechanism. So we selected drugs that specifically did not have that level of genetic evidence behind them.
And from Mike Hurwitz, sort of along that line, do you find it striking that every single one of your targets was wrong?
Yeah. So for the sake of time, for the sake of time, I focused on the ones that were where we discovered that the mechanism of action was incorrect. However, we did have a few examples where we could validate it. And I'm just trying to here, I'm going to show just one example of that now. So this is Nutlin 3A. This is a drug that's been reported to function through p53 activation, blocks the interaction between MDM2 and p53. We generated p53 knockout clones using Crispr. And when we did this drug sensitivity curve, we found that Nutlin has no effect on the p53 knockout clones while it kills the p53 expressing ROSA26 control clones. So in general, so this is what we would expect for a drug that acts through an on-target activity, you know, a huge delta between the target knockouts and the target expressing control clones. And we found a few examples of this.
Okay. And I think this is the last question from Karen Anderson. Did you make the serine mutant of CDK11 and show that the inhibitor was no longer effective in biochemical assays?
So we have been doing the biochemical assays through a CRO at the moment. We are not skilled in vitro biochemistry ourselves, and so we've just done it with through the CRO. And we'd be glad to launch a collaboration to investigate that, because I think that would be very powerful.
Well, I want to thank both Jason and Kurt. It makes me proud to have these kinds of presentations on my first day here. So thank you very, very much, and we'll see you all next week.